and good evening in other countries. Friends, today we have lecture number 18 by one of our very good friend, Dr. Umesh K. Reddy, who is a very good researcher. He is a professor of biology in West Virginia State University. Thank you, Dr. Umesh, for accepting our request to deliver this important lecture. He is going to talk on translational genomics for improvement of horticultural crops. I keep on seeing the messages about his good research, about his almost every month I could see his new research papers. He's really a very good research worker, and his research interests are in genomics, heavy metal stress, biotic stress related anthracnose, gummy stem blight and whiteflies, polyploidy, molecular changes pertaining to grafting in plants, evolution and domestication, breeding and qualitative genetics involving cotton, peppers, tomato, watermelon, blueberries, and pomegranate. They use Drosophila as model to study gut microbiome related to various phytochemicals. We are very happy to know that his Google Scholar citations are 2,897, a proud moment for any researcher. H-index 28, again a very difficult figure to reach for anybody and to dream of such H-index. I-10 index 43. So really, it's a great moment for us that we will be listening to him. He is a postgraduate from Tamil Nadu Agricultural University, and PhD he did from Osmania University, Hyderabad. He is editorial board member of many important international journals, like you can see the Comparative Genomics and Evolution, BMC Genomics, Plant Genomics and Biotechnology, BMC Plant Biology, Plant Genomics, Nature Scientific Reports, Genetic Mapping, Frontiers in Plant Science, Plant Genetics and Molecular Biology, Plant Molecular Biology and Physiology, and reviewer of many important journals of international repute. Friends, as you know, this series, the series of 75 lecture series, is being organized to commemorate the India's 75th year of independence, which is going to be on 15th August 22. And to commemorate that India's independence, we are holding this one lecture per week. Total lectures will be 75. And till date, we have organized lectures in different domains in agricultural research, genomics, biotechnology, motivational lectures also, like Sri Ravi Shankarji, lectures on general topics, also on environmental issues, lectures by our Honorable Minister of State about general policies, so like that. And we always request the eminent persons in different fields to talk on their areas of expertise. And today, we requested Dr. Reddy, Umesh Reddy, to deliver this lecture, which immediately and happily he agreed. We are fortunately the ARS batchmate also, and we know each other very well. Dr. Umesh, I know that there is a lot of time difference and may not be a very convenient time for you, but still you accepted our request and you devoted time to deliver this important lecture. Now I request you to please take the floor and deliver your lecture. Thank you, Dr. Umesh.
Oops. So can you all see the screen?
Yes.
Yeah. So just to, oops, it has not. It is my pleasure to accept this lecture. Dr. R.C. Agarwal has been my best friend for several years. And this is such a privilege, especially to give a lecture, as one of the speaker of 75 distinguished professors, the distinguished speakers. And I'm not that distinguished, I'm just a professor. However, getting this opportunity, I'm really indebted to Dr. Agarwal. So my topic, as I said, is translational genomics. Why it is translational? Because we have to get a lot of information from other crops, other model organisms into horticulture crops. Because not much investment has been done for research in horticulture crops. Many horticulture crops are orphan crops. By the way, I work in the West Virginia State University. There are so many policymakers here and general audience. So I would like to give a brief introduction for basic genetics. This is DNA is just 2 nanometers with molecule that is wrapped around these beads. So these beads are called nucleosomes made out of histone proteins. So this DNA is wrapped around these beads in this fashion. And then these are coiled coils, finally coalescing into total chromosome, which is about 1500 nanometers. This thing, 2 nanometers to 15 nanometers. This is all just like very high degree packing. So the whole DNA is not the genes. Genes are just about 5 to 10% max, of which only 1 or 2% are the exonic space, which is actually responsible for the phenotypic differences. So here are the beads that you have seen. These are histone proteins. So the DNA is wrapped around it. So just before transcription, by which the genes express, the process is called transcription. So the entire genic space will change. So first of all, this tightly wrapped DNA has to be loosened quite a bit, releasing its binding sites. Binding sites for various proteins like transcription factors has to bind. And then these histones have tails, you have seen. These tails have a lot of amino acids on it. So they all will undergo chemical changes like acetylation, methylation, phosphorylation. These kind of changes must happen before transcription. So when the genes mostly are silent, all of our genes, like in humans, if we have 30,000 genes, all the 30,000 genes are not functional at a given time. Only the genes that are required to function, they will function. Rest of them will be silent, sleeping. So that sleeping condition is called repression, repressive state, where all these histone tails that have amino acids are methylated. Methylation is kind of lock. So this gene is locked, but once this needs to be transcribed, then this genic space, all the chemistry will change. These tails will get acetylated and phosphorylated, and also methylation also for selectively on some amino acids is important for the transcription. And then the binding sites are open. Otherwise, these are tightly wrapped around these beads. Now it is open so that the transcription factors can bind it. Finally, the transcription happens. As a result of transcription, there will be messenger RNA and other RNAs that are important for translation. So the translation will happen in the cytoplasm. Eventually, what is the result of whole process is proteins. So I call phenotypes are like we have vegetables here, peppers, watermelons, and pomegranates, and melons. We have different phenotypes, fruit phenotypes, plant phenotype, disease-resistant phenotype. So all these phenotypes are manifested with the changes in the DNA. So genomics is once we sequence all of it, the global scale of study of the genes is genomics. And the epigenomics is how it is wrapped around these nucleosomes, how it is getting released, how it is getting methylated or demethylated. So all this comes under the epigenomics. And the transcriptomics is the transcriptional machinery. And once it is transcribed, all the messenger RNAs globally are called transcriptomics. Not only messenger RNAs, some of them are non-coding, some of them are small RNAs, microRNAs. All of them have regulatory importance. And proteomics is the global scale proteins, the whole proteins, if we can estimate and sequence, and we have these proteins, we can study and see the differences that which protein goes with what phenotype. And similarly, the metabolomics. So all these omics are to understand the phenotypes. So one important thing, this is not any hi-fi or rocket science, because the sequencing has become so cheap, inexpensive, and you don't have to have any equipment with you. You can just subcontract to any company and get your organism sequenced. So the training population is the population that breeders will have. Could be germplasm collections or their breeding populations that they maintain. So once you phenotype them, just one time you phenotype them, you don't have to phenotype again and again for subsequent years. Like the breeders make crosses, and year after year they have to worry about the phenotypes. But here in the genomic selection, once you precisely phenotype and genotype, and then you can identify the lines that have favorable allele combinations. So they are the better combiners. So this kind of better combiners, GEBV, are the models to identify the accessions or the breeding stocks with favorable allele combinations. So now we can make directory selections based on the favorable alleles rather than the phenotypes for subsequently. So this will reduce the time for the breeding, and also it is much more cheaper. Comparatively, like 10 years it would take for a variety to release using the conventional system. But this kind of genomic selection in combination with speed breeding, that is advancing the populations under the lights, under the LED lights, can be very economical and fast and precise. More like breeding is becoming more and more now technology rather than the art. So if we identify any deleterious alleles or susceptible alleles or any genes that negatively regulate any traits, now there are technologies to silence those genes. These are called genome editing tools. Here is the CRISPR. What we need to do is that gene-specific gDNA we need to design, and then once we put it in the CRISPR motifs, then there will be kind of, this kind of nicks will happen, this kind of degradation will happen in presence of this gDNA and this kind of nucleages that are surrounding to it. So this is the CRISPR technology using which we can silence genes. So if we don't like any gene, we can silence it. Or if we like any gene to be transferred from totally different organism or the wild species, can be easily done, can be easily performed using this technology. So now I would talk about my experiences in horticulture crops, especially how effective is this genomics to identify phenotypically discernible genes. So the trichome development is, trichomes are very important for plants because trichomes are the tiny hairs that are surrounding all over the plant, all right, on the leaves, on the stem, sometimes on the fruits. So what is the role of trichomes? A lot. Trichomes provide, the glandular trichomes provide kind of flavor or aroma kind of substances that can deter sometimes insects. And also trichomes are the physical barriers so that the insects will not attack the plant. Herbivory is kind of discouraged in presence of dense trichomes. And also the trichomes have a lot of role in phytoremediation. So we work on the trichomes in the watermelon. Especially this is a collection of watermelon, different kinds of genotypes. So to start with, what we did, we collected watermelons from all over the world, especially in the United States Department of Agriculture, GRIN, just like we have NBPGR in India, we have collections. So those collections were sequenced, and then we identified how diverse they are, the worldwide collections. So these are the cultivars, and these are semi-domesticated lines, and these are the wild species from which the watermelon evolved. Talking about trichomes, there are about 5 different kinds of trichomes. So the trichome research is very well established in tomato. And in tomato, they are deploying this kind of, especially acyl sugars that are from the glandular trichomes as insect resistance or insect deterrents. So there are different kinds of trichomes. Type 1 trichome is the glandular cell at the tip, but unicellular base. And type 2 are non-glandular trichomes, the spiny kind of trichomes with multicellular base. And type 4 are the glandular trichomes with several cells, multiple cells at the base. And then these are the non-glandular trichomes with unicellular base. So it is multicellular base, this is unicellular base. So there are different kinds of trichomes. Broadly, you can say the ones with glands and some without the glands. So all these are very important structures for the plant. So what we did, out of that 1,500 watermelons we selected, we identified highly diverse 170 accessions, and we grew them in the replications. At the 4-leaf stage, we have taken the petiole of the second leaf and then scanned under the microscope, and then used some publicly available softwares and estimated the density and the trichome length. And we have already SNP data since we have sequenced. So we conducted this genome-wide association study that is associating SNP polymorphisms with the phenotype. So there are, you see here, this watermelons, highly dense watermelon varieties and low density trichome watermelon varieties. And here is long trichomes and short trichomes. So these are all different kinds. You can see very long trichomes here, you all can see it probably. And here is the very short trichomes. So when we do phenotyping that precisely and do GWAS, then we can identify the candidate genes. So these are the genes, some of the important genes that interested us, especially this gene called tetratrichopeptide. Not much is known about this gene. So what we did, the Arabidopsis is a model plant. It is not any edible plant, it is just model for doing the genetic studies. So Arabidopsis also has all these genes. So Arabidopsis genes are, AT is Arabidopsis thaliana, 3 stands for the chromosome, and this is the gene number. So it all has all these genes. So what we did, we silenced some of those genes using tDNAs. Then what happens to the trichome, the normal trichome in the Arabidopsis that looks like this, the trichome that is branched. Watermelon do not have branches, but Arabidopsis has this branched trichomes, and you see how dense it is on the leaves. So in the mutant, once we silence this particular gene, the trichome shape changed. You see, it is degenerated. it lost polarity. So the branches are kind of degenerated. And then this kind of mutation happened because we knocked this mutant. So you see what happened here. The watermelon that do not have a lot of genomic information, we just did one-time sequencing and identified some polymorphisms, and then observed a trait very carefully and identified a gene that has exactly the same function that we are looking for. You see, so we don't have to have a whole lot of infrastructure. We need to have basic infrastructure to identify a candidate gene that interests us. So now this watermelon gene, we used on Arabidopsis. So we cloned watermelon gene and then rescued the function in Arabidopsis. All these are watermelon genes, tetratrichopeptide or TTL4 genes. And in transgenics, it shows clearly the function is restored. And this is the Arabidopsis gene. And then the mutant is rescued. And these are the overexpressed lines. Watermelon gene overexpressed in Arabidopsis, Arabidopsis gene overexpressed in Arabidopsis. So further now we can see what this mutant that we generated, the TTL4, will do. And we identified it has impaired cuticle. Cuticle is like wax-like substance all over the plant, and that deters insects as well as it provides drought resistance. Water will not transpire if it is densely coated with wax or cutin. You see, when the cutin is impaired because of this gene, it's not only trichome, but the cuticle is also affected. So this stain, this blue stain you are seeing, that is toluidine blue staining, that shows up blue if there is impaired cuticle. Cuticle is kind of dysfunctional. So here is the normal plant, the leaf of soil-grown 5 weeks old plant. And here is the mutant that has taken fully stain, indicating that the cuticle is impaired. And then this gene is very interesting, and how it is related to trichomes, because many laboratories in the United States are working on the trichomes, but how come nobody has identified it? That this gene is responsible for the trichome phenotype or density. So we wanted to see what genes it is interacting. So we used this gene as prey. This is the yeast-2 hybridization. It will have a bait protein and then prey protein. So the bait protein is the TTL4, and the prey protein is any protein we can use and see whether there will be expression of this reporter. So if it is expressing, that means these two are interacting with each other. So these genes are related to each other. So using this kind of yeast-2 hybridization technique, we have identified several genes that are interacting, and especially these TCP transcription factors are known to reduce the density as well as they have the trichome phenotype, and they are closely interacting with TTL4. And similarly, there are other actin remodeling factors that are also having trichome phenotype, known to have trichome phenotype. They interact with TTL4. So we had pretty much nothing, just watermelons, and we grew them, we sequenced them, we identified polymorphisms, and we tracked one trait and identified what is the gene responsible for the trait, and then understood how this gene acts, how it is interacting with the already existing genes. So much information could be generated in such a short time just by one single graduate student. So the tetraploids are very important in watermelon because not 99%, 100% consumer preference in the United States is seedless watermelon. So the watermelon is actually diploid. So they will give colchicine treatment and they make tetraploids. And once you cross diploid watermelon with the tetraploids, you will get triploid watermelon. Since triploids will have meiotic abnormalities, when the meiosis is happening, because of that meiotic irregularities, there will not be seed set. That would be seedless watermelon. So seedless watermelon is a huge industry. When I started this research, they used to say, well, 80% of our consumers prefer watermelons that do not have seeds. Now, pretty much 100%. And it is spreading all over the world. They prefer only seedless watermelons. See, the diploid, and once you double the chromosomes, that becomes tetraploid. You can easily say the tetraploids are much bigger. And similarly, the leaf size of tetraploids is larger. The flower is bigger. And the thickness, the stem thickness is more. And you see these trichomes, these hairs, what I have been talking, they are more denser, more stronger in tetraploids. But why the fruits are almost comparable, same size? So we wanted to work why these fruits are of the same size. And we wanted to understand what is this tetraploid superiority, how it is coming there in the tetraploids. So one of our grad students published a beautiful paper, and Plant Journal used it as a cover. She is the student, Marlene. Currently, she is in Max Planck in Germany. And earlier one postdoc, Tangasamy, also worked on the same and understood these fruits especially are becoming the same size because the extra genes, extra messenger RNAs that are produced are spliced through a process called alternative splicing. A molecular event, once the transcription is over, messenger RNAs are processed. In that process, some of the messenger RNAs are losing some of this. This is the diploid messenger RNA. And these messenger RNAs are spliced differently. And here is the messenger RNA is quite shortened. This intron became like exon. So all these changes happens in the fruits, finally reducing the messenger RNAs to just one copy. That's why the diploids and tetraploids are having the same size of fruit. And then we understood once the tetraploid watermelon, what happens, especially in the seedling stage itself, a lot of methylation happens. And then, the methylation, when we see it between the diploid and tetraploid, the hypermethylation is lesser and hypomethylation is more. So the demethylation also happens. In diploid, several genes are methylated. I said methylation is a repressive state, but in tetraploids that opens up. Probably that is one of the reasons for tetraploid superiority. And also nowadays we have a new sequencing called three-dimensional sequencing. Just like we see 3D movies, we have 3D sequencing. So in the nucleoplasm, when these chromosomes or chromatin material is entangled of different chromosomes, for example, blue is one chromosome, this red is another chromosome. So this chromatin interactions happens between the chromosomes. So earlier they used to think that whatever happens in the chromosome happens, that do not have anything to do with the other chromosome. But later they identified, no, the chromosomes also bang with each other in the nucleoplasm and they interact. The regulatory elements differentially function when the chromosomes interact. So we discussed already how the DNA is wrapped around these histones or nucleosomes, and then it is forming like chromatin loops. So these chromatin loops have these topologically associated domains. So these topologically associated domains having the regulatory elements, and they change. Sometimes these topologically associated domains will have two components. One is heterochromatin and euchromatin. That may be too much molecular biology for some of you, the policy makers, especially vice chancellors and the students here. But the heterochromatin is the silent stage and the euchromatin is active stage. So the genes switching into heterochromatin to euchromatin stage. So we identified that kind of switching, that is B to A, heterochromatin to euchromatin, or euchromatin to heterochromatin, how the switching happens across the chromosomes. So all these different processes, the expression, methylation, and how these chromosomal interactions happens between the different diploids and tetraploid explain why the tetraploids have superiority in comparison with the diploid. So one another important aspect in horticulture crops is grafting. We all know citrus is grafted, mangoes are grafted, you see, but the watermelons in India are not grafted, but in Europe and elsewhere, where you have this greenhouse watermelon production, the grafting is very important. Especially the watermelon, if it is grafted on the bottle gourd, lauki. The lauki has a lot of resistance genes, especially for the soil-borne diseases like fusarium wilt, and for several heavy metals and drought resistance. So many good traits are there in especially bottle gourd. If the watermelon is grafted on top of the bottle gourd, you can produce very high quality watermelon fruits, very large, and fruit size is increased and all. So we tested that. We grafted like that. In 2 seasons we tested. You see, this is the watermelon. Grafted watermelon is grafted on watermelon just to see is there any grafting effect. And then watermelon is used as scion and grafted on the rootstock of Lacinaria. Lacinaria means that is the bottle gourd. You see the fruit size increased once we grafted. Similarly, both seasons fruit size increased. If the Lacinaria is grafted on the watermelon, the fruit size is decreased. The bottle gourd size is larger, right? Similarly, the soluble solids, that is the sugar, watermelon is sweeter, but the grafting did not make any change. But very interestingly, if the Lacinaria is grafted on watermelon, the Lacinaria used is as a scion, then you see in the bottle gourd, the sweetness accumulates. The bottle gourd that is grafted on watermelon is sweeter, not very sweet like this, but it is a bit sweeter. If somebody wants to add a couple of spoons sugar in bottle gourd for making curry, if it is grafted on the watermelon, you don't have to do that, right? And also the rind pressure, the outer coat of watermelon is called rind. The pressure also increased. But Lacinaria is grafted, there is no difference on the Lacinaria rind. But watermelon rind, the pressure increased. Probably that can keep watermelons a little bit longer or keep them safer while transporting, especially in trucks and all. The watermelons bang to each other and get cracks. That kind of damage can be probably protected. And similarly, the rind thickness is same in the watermelon. But when the Lacinaria used as scion, its thickness reduced. We don't want a very thick outer coat. So what we have identified here is an amazing thing. When we grafted bottle gourd and watermelon like this, the genes from the bottle gourd are entering into the watermelon. The transcripts are the proteins. And then similarly, the genes from the watermelon is going down into the Lacinaria. So there is, genes are traveling across these grafts. So we identified 422 genes are traveling up or down. That was really amazing thing. And this paper is published and is highly accessed just to understand what is this long distance travel of the genes across these grafts. And similarly, the Lacinaria used as a scion and grafted on watermelon. And then the watermelon genes are entering into Lacinaria. And if the watermelon is used as scion, and then the Lasineria genes are entering into the watermelon. So back and forth, watermelon genes are entering into the Lasineria. So here we have identified 441 genes entering from the Lasineria to watermelon, and some 422 genes are entering from the watermelon. So this is very intriguing, and we wanted to see whether it really happens gene by gene. If we analyze, yes, it is. You see, the watermelon do not have this transcript. You don't have anything here. But watermelon is grafted on the Lasineria. In the Lasineria, it is there, this gene. You see, this band is the gene transcript. And then you can see it in the watermelon fruit. So clearly the genes travel once you graft. So that was understanding a little bit about the grafting and what is the implication of that movement. Currently we are working on it. So the most importantly in the genomics, we need to identify genes for the disease resistance. So here is the two tomatoes. This is resistant to anthracnose, and this is susceptible. These are the 7 days post-inoculate, pierced with the spores. And here is a little bit response. And here it is susceptible plant. The cultivar has huge anthracnose attack. So we made a cross, and we have a recombinant inbred line population using which we mapped the genes that are really responsible, the loci that are responsible for resistance or susceptibility. So these peaks signify, under these peaks, there are the genes, the causative genes for the anthracnose resistance. So what we did, since it is highly dense map, we could identify the candidate genes and how these candidate genes are in the population. For example, this gene in the susceptible plant, it has just one kind, but in the resistant germplasm has several SNPs. And that means this gene is undergoing positive selection, positive mutation to incorporate resistance in the resistant types. But in susceptible, it's only nothing is happening, purifying selection. But this gene is another important gene, and this gene has an extra SNP here. This is very important for resistance. That is lost in the susceptibility. You see the alleles of the same gene that makes it resistant and susceptible. So we did RNA-seq and identified that these genes are indeed having differential regulation, and then used this QRT-PCR just to quantify them. We identified the susceptible parent has high expression comparatively to the resistant. You see, this is the resistant parent. These transcripts are not expressing, but here it is very high expression in the green fruit, in the red fruit, all these different kinds of combinations. Clearly, these genes are expressing only in the susceptible plant. So what can we say here? The anthracnose susceptibility is because of some susceptible genes. So how to handle them? So somehow we need to use genome editing techniques and silence all these genes so that it would become resistant. We don't have to cross with the resistant parent, because resistant parent, in fact, is not having any special or unique genes in this case, in this particular disease. It is only the susceptible genes that are hitchhiking along with the quality or along with the breeding, the conventional breeding is happening. So these deleterious alleles that are causing susceptibility has to be just silenced. So currently a grad student is working on it. That is about it. And then the most important thing in the West Virginia, here we have a lot of acidic soils because of coal mining. Coal sludge gets contaminated all the agricultural soils. That's why here the soil pH is very, very low. Because soil pH is very, very low, we have aluminium toxicity problem. these heavy metals, especially watermelon, you cannot just grow in West Virginia because of this low pH and aluminum toxicity. The fruit is so distorted. So, but if we graft it on the bottle gourd, yes, we can definitely grow it. But one more thing, the vegetables that we are growing in heavy metal contaminated soils, throughout India, there are a lot of regions where you have aluminum toxicity problem and acidity problem, even in India. So the fruits and vegetables we are growing are contaminated with heavy metals. Though you are not spraying with pesticides, you may feel proudly that they are organic, but they are not. They are contaminated with these heavy metals, especially the horticultural crops like melons, fruits. They are consumed raw. So the effect of heavy metals are much more. So we started identifying the genes for the aluminum toxicity and acidity, especially using the Arabidopsis thaliana. Because now we understood that if we identify a gene in the Arabidopsis, it is translatable into the watermelon or any other vegetable crop. So the title of this talk, I named it as Translational Genomics, because the information pertaining to the genes and pathways, everything is translatable, right? So these heavy metals, once we stress the plant, we can use genomics, proteomics, ionomics, and all these different omics and perform the bioinformatics and identify integrated approach of plant response to these metals. And then we can further understand what we can do with this, either the transgenic approach or some kind of natural variation. We can look for the better alleles. First, we need to identify what are the genes that can help us to mitigate this stress. So again, talking about acidity, you see this, how acidity is spreading across the world. And this is going to limit food production globally in all crops. And in the horticultural crops, I told you, because we eat them raw, the effect is more. So it is a huge environmental constraint. And then under the low pH conditions, iron, copper, manganese, zincs are high, as well as aluminum is high. But the availability of nitrogen, phosphorus, and potash, especially you see the potash is very low under the low pH conditions. So we wanted to look for the genes that provide resistance for the aluminum toxicity or mitigate this toxicity. So we used Arabidopsis accessions. There are thousands of Arabidopsis lines that are sequenced multiple times. So we can identify, we know the polymorphisms, they are publicly available. We don't have to sequence even. We don't have to spend a dime. Just a good grad student can identify the genes in the Arabidopsis because a lot of resources are there. These are three different Arabidopsis lines, and you can see the response in the control as well as in the low pH conditions and the aluminum toxicity. This particular variety is having inbuilt resistance, whereas this particular accession, Arabidopsis ecotype, we call, not variety, ecotype. This ecotype is really affected a lot in presence of aluminum and low pH, and this is moderately affected. So this natural variation exists. So we performed association mapping because all these Arabidopsis lines are sequenced. So in just seedling stage, that is 5th day and the 10th day, we have identified there are 550 genes. They are responding to aluminum toxicity in treatment. And the 5th day, you see. And then we have identified about 1,751 genes altogether between 5th day and 10th day. And 403 genes are common. So they are working more 5th day as well as 10th day. Okay, so we have identified a gene. We named it as CRK because we have not yet publicly released this gene. See this CRK gene, the CRK is the gene. This plant is mutant. So CRK gene is silenced. If the CRK gene is silenced, what is its effect? In control, it is showing exactly like wild type. You cannot say that this do not have CRK gene because both are looking exactly same. But in low pH conditions, CRK is doing better than wild type. You see here how beautiful the growth is. And even the low pH and aluminum is combined, then this gene is functioning extremely well. So this CRK gene is a negative regulator for plant growth, because without CRK gene, the plant doing better, so it is a negative regulator. So if we silence the CRK gene, in fact, the crops, especially if we knock them out, the CRK gene in the crops, that can confer resistance to low pH and aluminum tolerance. So this is what translational genomics. Once you identify it in Arabidopsis, you can manipulate it in the crop plants. Or if you are identifying a gene in crop plants, you can study a little bit more carefully in the Arabidopsis to understand its mechanism. So here, the total root length, you can see the low pH and the aluminum, how the mutant is functioning in comparison with the wild type. And here is the total surface area of the roots. The mutant is doing better even under the toxicity comparatively to its wild type. And the root volume is higher. The number of chips on the roots is higher. So we could do this phenotyping of the seedling roots using a software. So similarly, the arsenic is a huge problem in this area. Arsenic causes cancers, Alzheimer's, diabetes, and many more things, including sexual incompetence. Arsenic is a very deadly chemical that do not have any benefit, very hazardous chemical. So a student was working, she published in Journal of Hazardous Materials. So especially the plants also have the chlorosis, necrosis, defoliation, reduction in growth, reactive oxygen species accumulation. So it is such a nasty metal, a very toxic metal. So using the same Arabidopsis research, we have identified some very good genes, especially for the arsenic resistance. And then this student tested 250 different mutants in this kind of media to identify this wonderful gene called F-box. So since this is published, you can read more details about it. So the F-box and the wild type, F-box gene is knocked off. So it is F-box gene is not there here. So this is the wild type. Even without F-box gene, it is not showing any difference with the wild type in control condition, a little bit difference. But in the arsenic, this plant is not doing well at all without this gene. So that tells us F-box is very important to mitigate arsenic stress. And here in different concentrations of arsenic, you are increasing the arsenic concentration, then the effect is much more. Without this F-box gene, this F-box mutant is really showing reduced growth. And then throughout the developmental stage, not only at the seedling stage, even in the adult plant, especially without F-box gene, the mutant is severely compromised. Its germination is reduced, and you see the F-box plant. So now we can say clearly the F-box is required for the arsenic tolerance. And then in the arsenic conditions, especially this is a stain called DAB stain that identify reactive oxygen species. So here you see reactive oxygen accumulation in the F-box comparatively to the wild type, indicating that RAS, reactive oxygen species, are much more here in this mutant. And then here is, inorganic phosphate is spiked. So once the phosphate is spiked, F-box is almost showing like wild type. And the proline content is higher in the mutant, indicating this has a lot of reactive oxygen species. This prolin accumulation is another mechanism to mitigate that. So identifying genes for heavy metals and then manipulating them in the croplands is possible to produce safer vegetables so that there won't be heavy metals in the fruits, and also they will produce higher yields. And then I'm not going to discuss a whole lot. This paper is widely accessed, and a couple of companies in Netherlands are using this gene to produce fruit firmness, especially in the melons, not watermelons, just melons like cantaloupes. What do we call it in Hindi? I forgot. Tarbuja, I think in Andhra, Telugu. So this melons, the fruit firmness gene we have identified using conventional mapping techniques. And also we collaborated with ICAR. Dr. Singh came to my lab, worked for 3 months. In 3 months, he published 2 papers. One is he identified microRNAs across the developmental stages of pomegranate. So using these microRNAs, we can understand various things like how to produce resistant pomegranates, because the fruit cracking and the bacterial blight are very nasty diseases, right? So once we know the microRNAs, we can identify appropriate ones and for doing further research. And then we do a lot of research on chili peppers. We collected thousands of chili peppers that belongs to various species. There are 4 or 5 different cultivated species of peppers: Capsicum annuum, Capsicum chinense, Capsicum frutescens, Capsicum baccatum. So all these accessions we have sequenced. And here is the baccatum, which is the ancestral species. This is the annuum, from where you have the bell peppers, Shimla mirch kind of peppers. And these are highly chinense, very hot peppers, just like Bhut Jalaukia and all. So these chinense peppers that are less pungent, much lesser than Bhut Jalaukia, are called habaneros in Americas, that are very famous here in the US and Mexico. And then we understood in the chinense, this round-shaped chinense are called habaneros. Even the Bhut Jalaukia, it looks like this when you cut it. So these are the ancestral ones from which these 2 types evolved. One is long peppers and one is this blocky peppers. So we did phenotyping using a kind of software, publicly available software, and we could estimate the fruit shape index. You see here the very small fruits just having 0.78 fruit shape index. And here the long peppers have the fruit shape index of 4.47, so 5 times more. So what kind of genetic mechanisms made these kind of peppers from this kind? And then here is another clade from which the habaneros and Bhut Jalaukias evolved. Scott bonnet also is another pepper. Caribbean scorpion is another pepper. So you can see very highly pungent peppers to very mild peppers within this group. So that paper is just published in Genomics. We identified genes for this fruit variation, and we validated using the Arabidopsis. So the details can be read in the Genomics. And a lot of, here is another gene that is very important for capsaicin. And then the same gene, here is the ankyrin repeat on the same chromosome, is important for capsaicin as well as fruit weight. You see the Shimla mirch is having larger fruit, but it is very sweet peppers, do not have any pungency at all. But the smaller peppers that have very high pungency because of this gene, You see the same gene duplicated, and one is important for the fruit size, another is important for the capsaicin. So, and then we have another great project in the laboratory. This interests many of you. Here is a fruit fly called Drosophila Melanogaster. So the Drosophila Melanogaster is a model organism. So if we can validate anything in the Drosophila Melanogaster, that is translatable to human, because humans and Drosophila, we share a lot of genes, lot of mechanisms. So if pepper diet, in the diet Drosophila food, if we mix the habanero peppers, whether it will do better to Drosophila or it will do harm to the Drosophila. So to understand that, we used these habanero peppers, the one just like Bhut Jalakia, but that the one that we fed do not have that much capsaicin. But you know, these peppers have not only the capsaicinoids, carotenoids, phenolic compounds, several vitamins and minerals. And I don't have to teach how the peppers have to be used in India, because all the food we eat will have some form of peppers. But in the US, only 5 to 10% of the foods will have this kind of pungency. And then a lot of studies already published. The genes, especially the brain-related genes, or liver-related, or fat-related, or pancreas-related, these genes are individually validated in the rats to understand what is the effect on the rats, and found the capsaicin has an effect for various different features here, you know. And then we wanted to see what happens. And again, whatever happens to the fruit fly, and we can translate this information to the humans. So, and also we had done quite a bit of research already, not here, okay. We did microbiome, I think, yes. So one student did microbiome, that is after feeding peppers to the flies, we dissected the gut of the flies and then sequenced, and we identified if we eat peppers regularly, our stomach will have very beneficial microbiomes like this. That's why probably we Indians are more immune comparatively to the Westerners, because of these beneficial microbiomes that are coming through pepper diet into our guts. So here we identified, here is the body weight in the control, that is do not have any peppers here. But there is significant difference. If the pepper is fed 7.5%, pepper powder is mixed in the diet of Drosophila, its weight is reduced. In the female, it is much more higher, about much more higher reduction in the weight. The triglycerides, one of the nastiest thing that predisposes us to the diabetics, is triglyceride percentage is really reduced, highly significant in female as well as male flies. But we did not find a lot of difference, though there is difference, but it was not significant about the glucose levels. So the peppers, chili peppers that we eat in our food reduce our body weight and reduce our triglycerides. And then we did metabolome, and we identified several important metabolites. Tocopherols are very important for vitamin E, and campestherol are important for vitamin D. And there are so many other metabolites are upregulating. A lot of metabolite, 6.1 folds metabolite comparatively to the control. And then some of the metabolites that are bad, that might cause diseases or that might predispose us to the cancers, are reduced, downregulated. So this kind of beneficial effects can be seen when we see the metabolomes, and also we sequenced its transcriptome. Millions of reads were analyzed to see the difference between the pepper-fed flies and the flies that are fed with that are not fed with the peppers. The difference between control and treatment. Here, treatment is we fed pepper, and control is no peppers. So we identified about 539 different genes uniquely regulating in the pepper diet flies. So these genes, if we compare, they are important for curing us from the Alzheimer's, Parkinson, and several cancers, cardiovascular diseases, and obesity. Pepper providing a lot of beneficial genes are upregulated, and then the harmful genes are downregulated, and helping us to be safe from these devastating diseases that we have today. So some of these genes have homologs in the human, so we can understand and what they are really identified, how important of them, especially this particular gene, TRPV1, that helps us to sense pungency. That also is modulate adipogenesis, so it reduces the body weight, obesity control. So this gene is there in the flies also. So all these genes are very important because they have homologs in the humans. We can understand how these genes function in fly much more faster and better than working with the humans direct. So my research is on to understand the domestication of crops, to understand the genes for the fruit quality and nutraceutical traits, and then to identify the genes for the biotic and abiotic stress, like especially the heavy metals, and then the effect of chili peppers in the diet. So this is about my research. And these are some of the grad students that worked with me and currently graduated. I think only one currently working in my laboratory. Otherwise, all of them currently left. We have different set of team. So my funding comes from different organizations, USDA, NIFA. NIFA is like ICAR that funds agricultural universities in India. Here, National Institute of Food and Agriculture is an agency of USDA that funds. And ARS is just like our ARS. Here we have ICAR together, but in US, ARS is separate. ARS has its own research centers, and NIFA is a funding body. We can write the grants and get funds from the NIFA. National Science Foundation also funds research. National Institute of Health also funds because we are working on the pepper diet effect. So that is related to human health. And we are working with some clinicians, clinical scientists to understand its effect. So if you have any questions, I can answer. Thank you very much for your patient hearing. And hope
Thank you, Dr. Umesh, for very elaborative and very educative, informative talk. I can say that those like me who did not have any background about this genomics also could understand some basic topics, basic concepts. So that was very, I can say, very useful for all kind of diverse audience. We can have, you can see the audience, they are vice chancellors. We have even Dr. P.L. Gautam, who was earlier Deputy Director General and Deputy Director General of this crop science and Vice Chancellor of G.V. Pant University. You can see from Konda Lakshman University, Dr. Nirja, she is also here. She is the Vice Chancellor in that university. We have from Bihar Agricultural University Vice Chancellor. So many diverse persons. And then you see a lot of other active research workers, the students who are present. And then we have another platform, ICAR 75 lecture series .webconevents.com, where people join without the video, so they can listen. So on this platform, we only allow only few important persons. So Dr. Reddy, there are some very basic questions, some specific questions. Our colleague Dr. Ravi Chandan is asking whether we can apply all these techniques in the cereal crops or not.
Yeah, of course, we can apply. Thank you, RC, for a great conclusion. And thank you for giving me this great opportunity.
So,
though it is midnight here, and I feel
Dr.
quite
A.N.
active.
Sharma, Dr. Umesh, Dr. A.N. Sharma is also asking some question.
Yeah, Dr. Sharma, very nice meeting you. We were, I think, together in the NAM.
Yes.
Are you the same Dr. Sharma? It
yes,
is
same.
Very nice. Yeah. So great.
Yes,
so
Dr.
yeah, anthracnose is a universal problem. And we started working with on sorghum anthracnose also in collaboration with Texas A&M. Recently, we have done some work. So it's a very nasty disease affecting several crops.
Anything else, Dr. A.N. Sharma, you want to ask?
No, it's okay. Just I wanted to know whether the silencing of susceptible gene will help in soybean also.
Well, probably that has to be validated. These genes can be expressed and see how they are working there.
Okay.
Yeah.
Thank you.
Thank you, Dr. Sharma.
Thank you, Dr. Sharma. Nice seeing you after a long time.
Dr. Renu Singh is asking, can you suggest how arsenic tolerance can be incorporated into rice?
Yeah, especially this gene, F-box gene, it is very interesting. It has homologs in rice. This can be overexpressed, and allelic variation can be tested in rice also for this gene. I, in fact, discussed with Dr. A.K. Singh, ICAR director, once that this gene can be really tested in rice.
Dr. Nivija, you want to ask something?
Really, sir, it's very enlightened lectures, sir, useful for the young scientists. I am very much thankful to the organizers on this occasion of 75 years of independence celebration, sir.
Thank you, ma'am.
Thank you. Thank you,
Sir, especially watermelon and chili related that genomic aspects.
Dr. Umesh, she is the only woman vice chancellor.
Oh, very nice. For which university, ma'am?
Konda Lakshman, Telangana State Horticultural University, sir. It is Mulugu near Shamirpet in Telangana.
Oh, I see. New university. Congratulations.
Thank you, sir.
Now, Dean from Merit, Sardar Vallabhbhai Patel University.
Sir. Thanks. Yes, sir. Can I interact?
Yes, please.
Thanks, Dr. Reddy, for a very good lecture and the work you are doing on various aspects. My query is that you mentioned that you have identified several genes which are responsible for abiotic stress tolerance in your studies. My experience in cereals is that the responsible genes are, minor in nature. They don't. They are not responsible for much phenotypic. They don't explain much phenotypic variance. So my question is, in your studies, what you have find, they are major genes or they are minor genes?
Yeah, this appears to be especially the arsenic gene as well as even the aluminium toxicity. These are quite major genes, commanding about 30% of phenotypic variation. So they are really, really major genes.
And for abiotic stress tolerance, could you find any major gene?
That's what for drought. We don't work on drought. I work only with the low pH and aluminium toxicity, arsenic, this kind of scenarios. But I need to see. In fact, one of our postdoc was working how these genes are affecting or playing role in other abiotic stresses, especially drought and salinity.
Thank you.
Thank you. Thank you very much. Any other question by anybody? If no, then Dr. Umesh, we immensely thank you from bottom of our heart that you gave such a elaborate talk, and you have really given a lot of information. And I think our students who are connected through the other platform, they can be immensely benefited. And if they are interested, they can further talk to you. They can further contact you in this respect. And as you know, we have a very interesting lecture of series. We have other two colleagues also, which Dr. Umesh has referred. So we are organizing their lectures also on very diverse research. So please stay connected with us on this platform, and always try to get a lot of new information for your research work and for many other areas in not only in research but also in stress removal or in corona-related topics or many other topics, which I can say. So we are devoting this particular platform for the overall information in multiple directions. So please stay connected. And thank you once again, Dr. Umesh, and thanks to all the audience, and especially Dr. Gautam, all the vice chancellors, our colleagues. I can see Dr. A.N. Sharma, we could meet after a long time, Dr. Ravi Chandran, and many others who have joined. Dr. Timothy also, I can see is present here. He's our Assistant Director General. So thank you all very much. Namaskar.
Thank you very much, RC, for giving me this opportunity. You all have a great day.
Thank you, Umesh. Thank you.
Bye-bye.
Thank you.
RC, thank you.
Bye. Bye.